First, congratulations for making it this far. You're still here. I'm glad to see you. And much appreciation for my opportunity to be here to present to this amazing group. I've learned so much over the last 2 days, and I'm glad to be able to do the best I can to tie everything together. So I am focused on milk secretory IgA and its impact on the commensal microbiota. I will be talking to you about this complex here, the commensal secretory IgA interaction, specifically in how commensals, or at least the few that I have been analyzing in its association with secretory IgA, the impact of this on commensal colonization, both in vitro and in vivo in a mouse model, and this investigation as to the ability of this complex secretory IgA and commensals to exclude pathogens. I don't have to go into much detail on secretory IgA. We have heard quite a bit over the last couple of days on this bioactive component. But just to remind you, this is found in abundance in both colostrum at about 5 mg per mil, and also over lactation age, which is seen in this chart, still maintains high concentration at about 1 milligram per milliliter with variation. This molecule is heavily glycosylated, and that's one of the things that I will focus on in this conversation. This is an image from Blaise Cordesi's review in Gut Microbes that does diagram nicely these N-linked glycans, heavily decorating the secretory component. The secretory component here, the heavy chain on both IgA1 and IgA2, and also extra O-linked glycans on secretory IgA1. These glycans have been analyzed quite extensively with Carlito Labria's glycobiology lab at UC Davis. This is one sample of this really amazing paper that looked at site-specific glycosylation and analyzed the relative abundance of these various N-linked glycans at each of these sites. This study was looking at 3 colostrum, so healthy women donating colostrum. And I did want to just kind of bring your attention to these sialic acid residues that we've heard discussed earlier today. And it looks like in colostrum that these glycoproteins are heavily sialylated, so with these sialic acid residues. And over lactation age, it does switch over to a more fucosylated, so more fucose residues on these molecules. So the function, and this is from a review by Nick Mantis's group from 2011 in mucosal immunology, that there are many, many functions already known of secretory IgA, and just to highlight a few from this nice review. We now know that not only are the secretory IgA molecules important in FAB-specific reactions, but these glycans also have a significant relevance in both immune surveillance and directly interacting with epithelial cells to reduce inflammatory response, including a reduction of the NF-kappa B pathway, an increase in the polymeric Ig receptor, which increases the amount of secretory IgA in the lumen, and even an increase in the tight junction binding proteins, so a promotion of barrier protection in these epithelial cells. We also know that with and without pathogens, so this is even with the presence of simply commensal bacteria, that we have a significant amount of sampling of luminal contents through M cell receptors that are glycan specific, so requiring the glycans on the secretory IgA molecule. And this interaction allows this reverse transcytosis of complexes of luminal antigens, again, both with commensal bacteria and also with pathogens, to these invaginations where we have dendritic cells expressing DC sign, which is a lectin receptor, and the mannose residues on these glycoproteins that are involved in binding to these dendritic cells and leads to immune tolerance, so expression of interleukin 10. We also know that, again, this FAB-specific pathogen bindings, this is really classic immune exclusion or pathogen exclusion, known for decades about the function of this molecule with that specificity. But also, many studies have now come out recognizing the glycans as also relevant in pathogen exclusion of this molecule, including a study looking at the S-fimbriated E. coli inhibited by those sialic acid residues on these glycans. Clostridium toxin has been shown to be inhibited simply by the glycans on the secretory component alone, so not even requiring the protein itself. And then a number of other mucosal pathogens are also excluded due to the glycans. so a seemingly non-specific interaction with this secretory IgA molecule. And finally, numerous studies now have come out looking at the impact of secretory IgA on the regulation of commensal colonization. And again, this could be due to both glycan interactions, as Blaise Cortese has demonstrated back in 2011, but also now some new studies have looked at this low affinity interaction of secretory IgA with commensal bacteria. And this is really where my studies are focused. So what the gaps in knowledge that our research groups, both Dave Mills and Bo Lauderdale, we've been looking at are first looking at simply the interaction between secretory IgA. This is looking at milk secretory IgA, so a very heterogeneous group of this glycoprotein interaction with select commensals, and looking at its increase in colonization potential. So I've investigated the digestion of these immune complexes, secretory IgA commensal complexes, both in vitro and in vivo. The ability for this to improve attachment, both again in vitro systems and also in a mouse model, and then investigating immune tolerance in the host. And second, looking at the complex itself. So are there any specific functions to this commensal secretory IgA complex? And this I'm looking at colonization resistance, so potentially the ability for this complex to help reduce pathogen binding, perhaps improve pathogen clearance, and of course have improved barrier function on the host. And this knowledge that we are gaining with these studies not only will help us investigate mechanisms as to what's happening in a natural breast milk system, but potentially in the development of a product that may improve the ability for probiotics, or it may improve administration of probiotics. This is looking at a possibility of creating some complex, perhaps with Rhamnococcus, Lactobacillus, Bifidobacterium, some very common commensal bacteria of the infant, and creating a complex with secretory IgA and these bacteria that when administered to the infant may help protect the probiotic from digestion in the infant's intestine, and improve colonization once this complex reaches the infant's colon, and at the same time, perhaps prevent pathogen infection with that FAB specificity still with these secretory IgA molecules. That's a big goal, and I'll show you how far we've come. So I've chosen to use this list of commensals. I won't go into much detail on this, but these commensals up here, these are Bifidobacterium species. Some are subspecies, including B. infantis, which is B. longum subspecies infantis, B. longum subspecies longum. These guys up here I've chosen due to A, their ability to grow on specific human milk oligosaccharides. The two I've chosen to use in my study are 2-fucosylactose, or 2FL, and lacto-N-neotetraose, or LNNT. These guys up here, these BIFs, these bugs, have also been extensively studied by the Mills Lab. So we have extensive genomic characterization of all of these organisms. And we have a lot of really fun side projects on looking at their abilities to, or their gene expression alterations with the presence of their particular growth substrate and alterations in binding to this particular glycoprotein of interest. I've also included 3 different lactobacillus, or lactobacilli here. Lactobacillus rhamnosus 7469. This is essentially the same species as an LGG. So I was looking at these as currently marketed probiotics, just kind of, again, looking at this concept of making this a translatable product. To obtain the secretory IgA, I just wanted to throw the slide up here in case you were curious. So this is the process of protein purification. I used donor milk for women who were over 3 months of lactation age, and collectively pooled together numerous milk samples. With this, I created the skim milk from here and flowed this milk through a capture select IgA affinity chromatography matrix, which allowed for really nice purification. This is demonstration with an SDS page and Western blot showing a really clear isolation of this protein from milk with virtually no contamination. So with this pure product, I then was able to answer some of these questions initially, looking at the interaction between secretory IgA and commensals. First, is there a concentration-dependent binding of secretory IgA to commensal bacteria? And second, how important are these N-linked glycans? So does the removal of these glycans lead to a reduction in the association of secretory IgA? Now, if you're familiar with IgA and secretory IgA research, you'll know that Blaise Cortese has attempted or has answered the second question to an extent. And so part of this was recapitulating his studies, and the part of this was really a novel investigation. And so what I found, and I used flow cytometry to analyze much of my secretory IgA association. In this plot, you can see Cyto9 as my DNA chelator, and I used an antibody actually that Jeff Gordon used also in his study. This is a Alexa 645 detection antibody for secretory IgA. This is an example of a flow plot, and this is Bifidobacterium infantis, so B infantis that you are seeing here, where up at the top you can see an increase in my concentration of secretory IgA, and hopefully you can see a very clear demonstration of an increased association of my bacteria with secretory IgA with increased concentration. So certainly evidence that this interaction is concentration dependent. And I wanted to look at this a little bit more detail with some of the top binders. So Lactobacillus rhamnosus, that LGG guy, B. longum and B. longum infantis, all three had very robust binding under the highest physiological concentrations of secretory IgA. And when I used variations of this concentration, you can see that these organisms performed a little bit differently with their interactions with secretory IgA. But interestingly, all of them seemed to reach a saturation and then plateau. Interesting to me, and perhaps those of you in the breast milk world, because, again, the physiologically relevant concentration of secretory IgA in breast milk is about 1 mg per mil. So this perhaps demonstrates that in that breast milk environment, we actually have lowered saturation of our commensal bacteria that are found in that breast milk community than they could be reaching. But more investigation is required for this. To answer the second part of this question, how important are these N-linked glycans at this interaction, I utilize a glycosyl hydrolase that's derived from B. infantis. This is endo-BI1. This cleaves that chitobiose core. It does leave behind a small residue, but it efficiently does cleave these glycans from glycoproteins, and analyzed my results of these digestions with SDS-PAGE, demonstrating a clear reduction in glycan isoforms on here. And found with immunofluorescence that these D-glycosylated proteins indeed have a reduced binding. This is showing a picture of lactobacillus with a native secretory IgA molecule, and then here's lactobacillus, same concentrations with a endo-BI1 treated molecule, where you can see significant loss of association of secretory IgA, at least with immunofluorescence and this detection antibody. So certainly some demonstration that the association of secretory IgA can be reduced with the depletion of these N-linked glycans. With this information, I also wanted to look at this function. So what purpose is this interaction? And Blaise Cortese, in one of his early studies, showed that the secretory component alone was protective with the digestion of the IgA molecule. And so with this, I hypothesized that perhaps commensal bacteria coated with a secretory-coated IgA may also be protected from in vitro digestion. And so I developed a model. It kind of took a lot of trial and error to evaluate this question. So I passed bacteria 2 times. The second time would be on the sugar substrate of interest, glucose for all bacteria, lactose, and then the preferred HMO substrates for some bifidobacteria. I checked the association, so I cultured these bacteria, bound it with secretory IgA, milk-derived, checked the association with FACScan, and then with this complex here, I then subjected this complex to gastric digestion conditions using pH of 4 with porcine pepsin, and then intestinal digestion with pancreatin at pH of 7. With this complex, I checked both live counting with CFU, serum dilution and CFU counting, and then I also used a flow cytometry with a cytotoxicity assay. And I evaluated this with all organisms of interest and found variations in the protective capacity of secretory IgA, but under all circumstances, with every bacteria analyzed, there did seem to be protective impact using secretory IgA. So this is log or fold change over these organisms digested without that protective antibody. in some cases reach statistical significance, in other cases very close to, but we'll be repeating some of these assays to truly determine this. But in all cases, if I did a regression plot using all different concentrations of Secretory IgA, I certainly saw a demonstrated correlation behind the protective ability for Secretory IgA to increase viability of bacteria post-digestion. So this was an in vitro model, and I wanted to see if this held true in a mouse model. And so in this first study, this is an investigation using BALB/c mice. This trial, we did a very short trial to investigate the survival, and I chose to use B. longum subspecies infantis. And again, this is one of these champion colonizers of the infant gut. So truly trying to tease out what's happening as best as I could with these infant commensals. For this trial, I collected baseline samples here to determine indeed that there were no B. infantis and really no bifidobacterium in these mice, followed by 3 days of gavage with B. infantis either associated or not associated with milk secretory IgA. I used that concentration that I had found was the highest saturation rate for this particular bacteria, and then collected fecal samples 1 day post-gavage, necropsied on the second day, to determine the amount of bacteria that essentially were able to survive that passage and persist for at least a short period of time. And what I found was indeed that when B. infantis was pre-associated with secretory IgA, and this was milk IgA, that I had almost a complete log-fold increase of bacteria that were able to passage through that intestinal content and be shed through the feces. Now, I will make one caveat that was one of my colleagues brought to my attention. This is fecal bacteria, so I cannot say with this plot alone that this demonstrates persistence in the intestine. We do have biogeography studies that certainly do show there is still almost the same, not quite, about 10 to the 5th CFU in almost every part of the intestine post-digestion, so at that day of necropsy. So I cannot completely say if this is due to just shedding of the bacteria or if it's evidence of survival of the bacteria. But nonetheless, again, with biogeography studies, I can say that these levels still did seem to be much higher in those mice that were provided that B. infantis that was pre-associated with IgA. So certainly, at least in this study here, we do see some evidence that secretory IgA can improve that survival of commensal bacteria post-digestion in these BALB/c mice. And the last part of this initial investigation with just commensals and secretory IgA was to look at this ability for these commensals to bind to both an in vitro co-culture system of human colonocytes and then also in BALB/c mice again. So for the in vitro trials, I, again, through much trial and error, came upon this concept of utilizing HT29-MTX cells, which have a very high goblet cell production, so I get a very significant mucosal layer that I think better represents what's happening in the colon. And I used Keiko2s also, so this is a co-culture. For these ones, I again used B. infantis, and I wanted to just do an evaluation of persisted B. infantis after a short period of time sitting on these colonocytes and look at any expression changes in the colonocytes themselves. And again, what I found was as I increased the association, and this is pre-association before I put the bacteria onto the surface of these colonocytes, so as I increased that percentage of population associated with B. infantis, I also saw a very clear increase in the amount of bacteria that were remain bound to the surface of these colonocytes, even after washing many times with PBS. And just to plot out, I took that slope and gave you a clear number. So for every 1% increase association for this bacterial population, you see about 10 to the 4th CFU increase binding capacity. So it seemed to be a very robust binding capacity with secretory IgA. And then again, gene expression studies on these co-cultures. When B. infantis was provided, and this is log fold or fold change over baseline with PBS alone, there was a significant alteration in a number of different genes involved in inflammation and also in tight junction binding proteins. And when secretory IgA was associated with B. infantis prior to that interaction with these co-cultures, essentially it brings things back to baseline. If you did look over B. infantis without and B. infantis with secretory IgA, you could say that there was increased tight junction binding, increased mucosal production, seemed to be beneficial for the introduction of this commensal to first have the Secretory IgA association prior to that interaction with these colonocytes. And then again, moving this into an in vivo system, so looking at this inside of a mouse. This was again in biopsy mice. You'll see a very similar timeline here where we wanted to collect baseline samples to ensure that there was no B. infantis in here initially. Gavage for 3 days with B. infantis. And here we have the beginning of my complex slide. So we have 4 different groups. 2 were provided water, but 2 were provided with 2-fucosyl-lactose to really increase the ability for that B. infantis to find its own little ecological niche and survive. We collected fecal samples day 4, day 6, day 8, and then necropsy done day 9 to look at persistence of B. infantis. And so what we found, I'll break this down just a little bit, when the mice were provided with water and B. infantis alone, there really was no detectable levels of B. infantis in that mouse on day 6, day 8. They were gone, washed out. When the mouse was provided B. infantis with that 2-fucosyl-lactose alone, we now were able to detect levels of persistence over all days of this trial. So in this case, it looks like that 2-fucosyl-lactose alone will increase persistence of this B. infantis. Now, this is also true if you've been following any of Dave Mill's work. We've also looked at this with B. longum, SC596, and found actually the same thing. So when these HMO-consuming bacteria are provided essentially their little suitcase of food, then seems to be that they can persist in even models where they normally would not. But of course, my question is, does Secretory IgA do anything to this complex? So in this next group, this is B. infantis provided pre-associated with Secretory IgA, and this is, again, back to that water model. So whereas we initially saw no persistence with B. infantis provided alone, now with just that provision of B. infantis complex with Secretory IgA, you see them. They popped back up again. So it seems that Secretory IgA did indeed increase the persistence of B. infantis, and this was over or almost to similar levels as just the provision of 2-fucosyl-lactose alone. So let's combine both of these together. What happens when we give these mice both 2-fucosyl-lactose and B. infantis associated with Secretory IgA? It seems to be a combined effect. So with these mice provided both 2-fucosyl-lactose and pre-associated B. infantis, this spikes the levels up, and I didn't put my significance on here, but this group is definitely statistically greater than or higher prevalence than any of the other groups together. So a clear demonstration of the combined effect of both that specific HMO and Secretory IgA. And again, just to kind of bring this back to what we're talking about in this whole group, right? This is the infant. That we're looking at something similar to an environment where the infants are exposed to commensal bacteria that perhaps are associated with Secretory IgA, if you're looking at this in the breast milk environment, and of course have the presence of many different HMOs in that same environment. So perhaps this combined effect does really improve the ability for commensals to get into the infant and to colonize inside. So in summary for this first section, Secretory IgA, I can say, certainly increases the colonization of select commensals. This was not true with every commensal evaluated, but certainly with B. infantis and a few of the others that we evaluated, including increased protection from digestion, improved colonic retention, and also the improved host response. So with this information, I wanted to then look at the function of this complex in pathogen exclusion. We actually collaborated with the Wadsworth Center in New York with their model of Salmonella invasion. So we used Salmonella typhimurium, and we wanted to investigate essentially the functional cross-reactivity of a monoclonal recombinant Secretory IgA called SAL4. This is specific to non-typhoidal Salmonella, and complexing this with B. infantis and looking at the prevention of this enteric pathogen, both in vitro system and also in these biopsy mice. So I'm just going to take a minute and explain this pretty cool model that we adopted from the Wadsworth Center. They generously provided these 2 strains of Salmonella typhimurium. One is the mutant strain, and one is a wild type. Both have kanamycin resistance, which allows us to grow these on selective agar, especially in a mouse system, so we can isolate these guys out. But the wild type has this O5 antigen that is specific to the SAL4 secretory IgA antibody. This has been extensively studied in the Mantis lab and also with Blaise Cortese, and shown to reduce the ability for this Salmonella typhimurium to invade and persist inside an animal model. And then this wild type has a deletion in a gene that's encoding the acetylation of the O5 antigen. So in this case, we have no SAL4 interaction, at least not a specific SAL4 interaction. I do have evidence that this still does bind to this guy, but does not seem to be due to that specificity of FAB. These guys also have that LAC-Z gene, a reporter gene that allows them to turn blue under blue-white screening agar. So again, a good way for us to differentiate one to the other. We chose, therefore, to use a competitive index, and this is both for our in vitro models and also in our mouse models, to evaluate the impact of the specificity of the SAL4 antibody. And for these competitive indices, we just did a wild type divided by mutant bacteria, and this is looking at those that were able to invade colonocytes or able to migrate to the Peyer's patches in a mouse model. And the idea here, a competitive index of 1 essentially means an equal number of both the wild type and the mutant. And a competitive index of anything less than 1 indicates SAL4 specificity, where we had a fewer number of the wild type and no change in the mutant. So we were looking at this as our baseline marker to evaluate the effectiveness of SAL4 specificity, and also to get away from the inherent variability in mouse models to look at Salmonella invasion. For our in vitro models, we used, again, actually for both, we used B. infantis. And for our treatment groups, we have 4 groups here. First is the Salmonella. This is our positive control, just Salmonella alone. Second is the Salmonella mixture. So we had equal molar concentrations, essentially, of the wild type and the mutant. And in this negative control, we have our SAL4 specific antibody that should be able to inhibit the wild type but not the mutant bacteria. And then our question mark, so B. infantis provided as a preconditioning to these colonocytes prior to Salmonella invasion or infection. And then finally, this truly a question mark here, does this complex of SAL4 with B. infantis have any specificity at preventing invasion of Salmonella? And what we found. First, just looking at total wild-type bacteria able to invade a colonocyte mixture. This is our co-culture system. This is our positive and negative control. So we do indeed see a reduction with that SAL4 specificity of our wild-type bacteria. When B. infantis was provided with no SAL4, we still saw levels that were the same as in our positive control, which was expected. And then now with B. infantis that was pre-treated or pre-associated with SAL4, we saw indeed, there was a reduction in the wild type, not exactly to the same levels, but still statistically significantly reduced for the wild type bacteria. And again, to measure the specificity of the SAL4 antibody to see if this really was that antibody that was doing this reduction bound to the surface of this commensal bacteria, to look at the competitive index. Our positive control had no difference. This is just Salmonella provided alone. No difference in wild type versus mutant bacteria. When provided that SAL4 preconditioning, the wild type was inhibited and the mutant really was not. So we had a significant drop, about 50% less wild type bacteria than mutant. When B. infantis was provided as a pre-treatment here, we saw back to baseline, no difference in wild type versus mutant. And now the question of selectivity with that SAL4 antibody. Indeed, we saw that when B. infantis was pre-associated with that specific SAL4, the specificity of that antibody was still there, was able to selectively inhibit the wild-type bacteria over the mutant, indeed to even greater levels than when SAL4 was specifically provided to that complex. So this was a great indication that our hypothesis held true. B. infantis complex with secretory IgA was effective in reducing Salmonella invasion, and indeed was effective with that specificity. So the FAB-specific binding was still present even when that B. infantis was associated with the secretory IgA. Looking at gene expression, this was just in the mammalian colonocytes. So we saw that B. infantis alone was able to reduce interleukin-8 production. This is a pro-inflammatory cytokine. This is Salmonella, and this is B. infantis. It did not seem to be Secretory IgA had a significant impact here. But when Secretory IgA was pre-associated with B. infantis, at least some tight junction binding proteins were increased with the presence of Secretory IgA versus when it was not. And then finally, I'm almost done, finally looking at this, moving this into a mouse model. Again, the same kind of model here where we had 3 days of B. infantis gavage. And in this case, we wanted to look at the ability for that specific SAL4 response to Salmonella. This was our first control group where we had Salmonella provided alone with no B. infantis as our negative control, Salmonella provided with that SAL4 as our positive control. I'm kind of flipping those around. And then these mice were provided that B. infantis with or without SAL4 and infected with Salmonella 1 day or 3 days post B. infantis gavage. And again, we had different groups here, provided either water for these control groups here, or when B. infantis was provided, we gave these guys 2-fucosyllactose to improve persistence of B. infantis. And what we found, first, just looking at total invaded Salmonella with both the wild type and the mutant, no difference. No difference in any of these groups with total Salmonella invaded into the Powers patch. But again, looking at competitive index was more our question. With the competitive index, when there's no B. infantis, just Salmonella alone, our competitive index was close to 1. And then when that Salmonella mixture was pre-associated with SAL4, we saw that competitive index drop as expected, showing you the specificity of that SAL4 antibody against the wild-type bacteria. So this was what we were comparing all of our treatment groups to see if anything showed the same level of specificity and reduction in invasion. Our first group, when we just infected with Salmonella 1 day post-gavage, there really was no difference from that initial competitive index. But here, when we had B. infantis that was pre-associated with SAL4 and we gavaged with Salmonella 2 days post B. infantis, we did actually see a reduction in the competitive index, indicating that this SAL4 that was pre-associated with B. infantis in the gut of a mouse 3 days after the presence of this or the introduction of this was able to selectively reduce the wild-type Salmonella bacteria from invading the Powers patch. So indeed, we did see the same or similar results as we saw in vitro, that this complex does indeed prevent that Salmonella invasion into the Powers patches, similar to the provision of that SAL4 alone. So again, this complex here, this idea of the function of this complex, we certainly can see that it was functional in preventing enteric invasion. And then overall, just looking at this, the concept of this function, increase the persistence of the commensal bacteria and perhaps prevent other enteropathogenic infections. And with that, I just wanted to say a great thank you to my enormous Secretory IgA team that I've collected over time, and then both the Mills lab and the Lauderdale lab where I do all of my work. And with that, I can open this up to any questions.
Thank you very much, Vanessa. Questions from Dr. Ogra?
Thank you so much. It's a wonderful study. And I promised myself I'm going to learn more here today. Can you define for me what you call commensal?
That's a good question. So we've chosen, and I use the term commensal most of the time in this conversation. So commensals are those that are derived from the infant, essentially these ones, the infant mammalian colonic tract, and able to essentially persist inside that organism. So these commensals, the ones that I've chosen here, were all isolated out of a healthy infant and used because we see them in the human population.
Because I don't think human body recognizes that this is a bad bug, this is a good bug. The bugs don't know they are bad or good. It depends upon how they have been, they have adapted to the human host over thousands of years.
Indeed,
really.
Indeed.
So the question is the specificity of the system here. If you were to take a pathogen, what you call not a commensal, do the same thing, will it do the same thing as Lactobacillus does?
So I've looked at this a little bit, and we looked at association with milk Secretory IgA, with Salmonella, with EHEC, with Campylobacter, and do see that it can bind to these organisms, and it seems to be more preventative than it is increased increased persistence. So at least in the in vitro system that we're looking at, when I pre-associate Salmonella or pre-associate EHEC, for those, of course, it's a little bit different model. With Salmonella, I can see that actually milk secretory IgA alone is inhibitory to invasion of Salmonella to these colonocytes. And with EHEC, I see a reduction in adhesion of EHEC when pre-associated with secretory IgA derived from milk. So it does not seem to be doing the same thing. It does bind non-specifically, but...
See, that's what I'm a little concerned, whether this is a question of the model you allude. Because Salmonella is not a mucosal infection as such. It's a lymphoid cell infection. And it simply binds to the epithelium and then gets into bloodstream.
Yeah.
And the dose for Salmonella is pretty large and pretty variable, tremendously. If you were to use something like Shigella, would you be seeing something different? Or if you were to use a pneumococcus, which is coated with IgA, and give it instead of your commensals, would you see the same effect?
I don't know. I think that we should investigate that and see if we do see a similar effect. I mean, I did look at, as I said, 3 different enteric pathogens, and all seem to be holding true that milk secretory IgA still seems to be somewhat protective, even with Salmonella. Although you're absolutely right that the epithelial cells are not its end goal. It must still pass through those epithelial cells to get into their invaded tissue, and since that is actually what we're looking at, is just the prevention of that invasion.
But there's no specificity for the secretory IgA for the commensals which are used.
Yeah, it doesn't
look
like it.
Any IgA probably could do it.
So how does it know is the question?
How does it know it?
Yeah.
You know?
That's a good question. I mean, and of course we're looking at, in general, we're looking at gram-positive versus gram-negative here as well. All of the pathogens that we're looking at here are gram-negative, and the commensals, all of the ones we've chosen are gram-positive. And even Blaise Cortese, originally when he began this investigation, saw a difference and he used E. coli, the Nissel version, so this commensal E. coli, and saw that it did not perform the same as the gram-positive commensal bacteria. So perhaps the specificity is more due to just the gram nature of the bacteria and not necessarily to its pathogenic nature.
I think you did a fantastic job, the youngest and the last speaker for this conference.
Most of you stayed awake, thank you.
But I would really look at some of the quote unquote pathogens versus commensals.
Yeah.
Because they may function quite differently.
Yeah, they might.
And there are earlier data to suggest that IgA can bind meningococcus or gonococcus and prevent it from lysis by IgG and IgM also.
Yeah.
So there may be other mechanisms by which you can affect either uptake of the antigens or prevention of the antigen destruction by the host.
Yeah, one thing that I have not had the opportunity to do yet is do any mechanism-based investigation. I mean, the SAL4 antibody has been thoroughly investigated by the Nick Mantis' group and others, and looking at its ability to actually alter the bacterial cell itself, changing its flagellum motility, et cetera. And I actually found a reduction in even the wild type with SAL4. That specific antibody is still the mutant bacteria was still able to bind to that specific antibody and still reduce its invasion. So something else is at work besides that specific interaction with these antibodies. It'd be a very interesting study to do more mechanism-based research.
Thank you.
Yeah.
Dr. Walker?
Very nice presentation. I know that David Mill's favorite organism is bifidobacteria infidus.
Yeah.
And I think he associates that with breast milk itself. So the question I have is, have you incubated that organism with breast milk to see if you get a complex that would occur in a manner similar to what you showed in vivo?
We have talked about that, but we've not done that yet. In part, no, I've not done that yet. I think I will. It has been done, just milk and commensal bacteria, and just pulling it out.
It has been done?
It has been done.
Yeah.
Because that would be a more physiologic approach.
Certainly.
Yeah.
But there are so many other factors in milk that could alter the binding of, BIFs to secretory IgA. There are different physiological functions or genetic alterations that happen with that bacteria in that milieu of milk. So it would be difficult. It would be an interesting just look to see what happens, but to figure out why it's happening, it would be a big question. And in part, not only is this study looking at what's happening in milk, but also what could happen if we took this out of that milk system and we're trying to introduce probiotics to maybe preterm infants, right? And making sure that we are not stimulating a robust inflammatory response with these guys, because obviously the infants don't know what's good and what's bad. So if we can protect these on the way in, it might be beneficial.
Yes, a question from Dr. Lifshitz.
Yeah, indeed, very nice presentation. My question goes a little bit to carry this type of finding to the human. And I would assume that the majority of the secretory IgA is produced in the small bowel. Is there evidence that the secretory IgA is alive and working in the human colon and not degraded by the microbiota?
Jeremy, no,
Actually, the density of cells producing IgA in humans in the colon is much greater than in a small intestine. So probably there is more IgA in secretions of the large intestine than a small intestine. So, why is that? I don't know, but probably because of the local stimulation, expansion of cells by the... If you look at the distribution of bacteria in the gut, in the stomach, you have basically nothing, or maybe a few hundred bacteria. Duodenum, few hundred thousands. And you go all the way down, and suddenly you reach the large intestine, and you have to 10 to the 12 bacteria per milliliter or gram of the stool. So it's a tremendous amount. And I don't know whether anybody, Ray, you may remember, co-culture or culture of intestine, small intestine versus large intestine. Tom Tomasi did something like that, but
Small intestine does produce more IgA, but there's a lot more space.
no, sure, because of the length, but per unity of the, I showed the slide of it from Per Brancek, there are more IgA producing cells per square millimeter than in a small intestine. There's no question about that.
But also,
But
IgA
it's,
is functional in the large intestine, regardless.
Sorry?
it is functional.
It's still functioning in the large intestine, even if it's produced in the small.
Oh, sure,
Yeah,
sure.
absolutely.
That's why milk may protect, because it passes through all the way.
Yeah, there's
Is that
certainly...
answering your question?
There's absolutely evidence of bioactive secretory IgA in the colon. I mean, there are numerous studies. We're looking at animal models with deficiency in secretory IgA or secretory molecules, and you see that what normally you see in the colon, with it, which is the colonization of that outer mucosal layer, that those bacteria are able to really penetrate that outer mucosal layer and enter either into the inner or even to the colonic cells without secretory IgA or secretory IgM present. So certainly it is not only found, but most important in the colon.
One of the things I might have suggested to look at polymeric IgM also,
Yes.
especially in the absence of secretory IgA.
Absolutely.
And it may do as good a job.
It might.
Maybe better.
Yeah, yeah, I will.
Dr. New?
Have you given any thought to maybe coupling the SIgA to eco-groups, the types of things that Jeff Gordon has been studying with the Malawian children, where you take a consortium of bacteria and you take them from a set of healthy children, and those you then would add to malnourished children.
Yeah.
But if you could provide the most efficient route for that eco-group, it would make a lot of sense with what you're saying.
Yeah, absolutely. So we've not followed, we've not in our laboratory done the same levels of investigation as Chef Gordon does in identifying groups of bacteria associated with a particular phenotype, but the idea of creating a concoction, essentially, of different organisms has been something that we would really like to look at. And in particular, I mean, we looked at lactobacillus and bifidobacterium in part because it's theoretical that lactobacillus are more small intestinal and bifidobacterium are more colonic. So if we can create something that might essentially be beneficial all the way down the intestinal tract, especially if we're looking at prevention of enteric pathogens. So many are small intestinal, and BIFs might not have an impact on the small intestinal pathogens, but lactobacilli might. So it might be a really interesting investigation to take this concoction of both small intestinal and colonic bacteria, associate them all with secretory IgA, and sort of put them into a model with both small intestinal and colonic pathogens to see if it does the same thing. Yes, ma'am.
In case of IgE deficiency, IgA deficiency, if IgM is covering also similar
Yeah, I think there was I did not do the study, but I think there was a paper looking at Secretory IgM association, right? I forgot who did that analysis.
No, there are antibodies, yes, that has been done, but effect like you have examined, epithelial cells, that has not been done.
Ah, okay, yeah. But I do know, yeah, they do associate certainly with numerous members of the microbiota. Mm-hmm. Okay.
If no further questions, thank you very much, Vanessa. And with that, I would like to thank